Hello student, today I am going to explain about the pedigree analysis. Now question is, why we'll do the pedigree analysis? That analysis, first of all, what we'll analyze? We'll analyze the diseases, or you can use syndrome, diseases, or you can use syndrome. Okay. Now what are the diseases and what are the syndromes we are going to analyze through this pedigree? And another thing, let me tell you that pedigree analysis is one kind of assumptions, or you can use the probability. Probability. So probability of the syndromes in next generations we can find out, or we can rule out. Now here at, I think you have finished the topic about the mutation, and we know what mutation is. What mutation is? The sudden change in the chromosomes, or you can use gene, the base pair, changes of base pair, changes of base pair in DNA, or morphological changes of chromosome. Uh, we know about the Creutzfeldt syndrome, about the Creutzfeldt syndrome on fifth numbers of chromosomes. If it will delete it, then one disease will called, or not disease actually, that syndrome will cause, which is called as Creutzfeldt syndrome. Another thing is, if number of chromosomes will change, then also it comes under mutations, because due to the mutations also number of chromosomes, number of the part of the chromosomes may deleted, or number of chromosomes may added or deleted. So as you are in class 12, obviously I don't need to explain about the autosomes and sex chromosomes. We have what? We have 23 pairs of chromosome, out of which 22 pairs autosomes, and XX in case of female and XY in case of male, it will be one pair. So by this only we'll be having 46 numbers of chromosomes, or you can use as a 23 pairs of chromosomes. Now if anything changes on 23 pairs, if anything changes on XX or XY, then this type of changes which cause syndromes or disease will be ruled out by the pedigree analysis. Now you may ask one question also, why we cannot use the conventional method? Conventional, why will not use, let me write, the conventional method. Conventional method which was discovered by Mendel. The Mendel was mathematicians, we know. Mendel actually worked on Pisum sativum, or you can use pea plant. So we know what, in case of pea plant, there are contrasting characters actually. I think as it is the chapter ending topic, obviously you know about the why that Mendel has chosen the pea plants. It can produce the offsprings within short period of times, having contrasting characters, and multiple numbers of progeny can be formed. So let's see, suppose in case of F2 generation, normal crossing I am telling. In case of F2 generation of dihybrid cross, I think you may recall the ratio is 9:3:3:1, isn't it? This ratio is phenotypic ratio. If you sum up, it should be 16. So 16 numbers of offspring will produce. So on the case of human, in case of human, to rule out the problems or disorders after 16 numbers of progeny, it is never possible, because we know what, amdo, amarado, or you can use one terms. Nowadays also it has converted into neutral family. So हम तो हमारा एक, that means what, we are two, the couple will be having one kids. So on that, or on them, that type of conventional method cannot be used. For that only, that probability which will be caused by the changes of chromosomes and autosomes can be ruled out by the pedigree analysis. I think you have understood somehow that pedigree analysis actually helping us to identify the problems, to identify the problems in the couples also for their better futures, isn't it? Now let's see another thing. What will happen if number of chromosome will change? Suppose, I think some of you, or I think as it is the part ending part, you may heard actually the two terms about the aneuploidy and euploidy. So aneuploidy means what? If number of chromosomes, if number of chromosomes, you can use plus minus 1 or plus minus 2 up to this much will change. Then the condition is called as aneuploidy. Suppose in case of aneuploidy, I can take one example of autosomal disease. One is suppose Down syndrome. Let's see. Uh, in case of Down syndrome, on 21st numbers of chromosome, there will be, suppose this is 21st pair of chromosome, there will be extra. There will be extra chromosome. Now this condition is called as what? Trisomy. Okay. And you will see in case of this one in Down syndrome. Now, will the Down syndrome can be analyzed by this pedigree analysis? My answer will be no, because the Down syndrome, it is not a Mendelian disorders. It is actually chromosomal disorder. Mendelian disorders will be what? Mendelian disorders will be what? The disorders which can be ruled out by the pedigree analysis. Now, question is, what type of diseases or syndromes will be ruled out by the pedigree analysis? This is question mark. After a few minutes we'll tell. Now let me explain about the euploidy. Now probably you may heard about the euploidy when the set of chromosomes, suppose we'll have what? 2n numbers of chromosomes. 2n numbers what? Yeah, 2n is equal to what? We'll have 46. So n set is how much? How many? Here will be 23. So in case of us, when n will be multiplied into 2, n will be multiplied into 2, then it will be converted into 46. So if in normal persons, if somebody will ask what is the conditions or ploidy conditions in case of normal human, then it will be what? Euploidy conditions. Now another term which is very very important you should know before doing pedigree analysis, which is called as polyploidy. Poly means many, obviously, when many set of chromosomes will be increased, which is actually not usually. It is actually unusual conditions it may occur. So n if you will multiply into 2 into 3, what will have? Suppose that 23 3. So it may convert into 69. So this 69 in case of human, if it will occur, so that baby will be died inside the womb. Maybe the research told that baby will be dying inside the womb. So miscarriages may cause. So you can not only in case of human beings, in case of plant also these things occur. So nowadays people what do? People actually do induced mutation. It is also, as it is a part of mutation, the people actually nowadays people becomes more what I can say, more advanced, and to mm, gain much more money, they are doing a lot of things nowadays. What they are doing? Induced mutation. Induced mutation means what? They are creating the mutations on the plant, so that only number of chromosomes, or not number, actually set of chromosome will be increased. Set of chromosome will be increased. Set of chromosome will be increased, and artificially you can use artificially, and farmers will be benefited. Farmers will be benefited. So actually this is not a class to explain about the induced mutation. I just give you some knowledge before starting the pedigree analysis. Now a few minutes ago I told you, as Down syndrome, it is not a Mendelian disorders. So here you have seen or you have heard or what I don't know, that whether Down syndrome is autosomal dominant or autosomal recessive. That type of word no teachers will not use. Because of what? Because we are actually trying to find out the Mendelian disorders. In case of Mendel's, the Mendel's only use of either they're dominant and recessive, isn't it? So that is why we are going to know some diseases, 2 or 3 diseases, which we'll be using to, or which are actually that type of questions may come. Suppose hemophilia, whether it is X-linked or X-linked dominant or X-linked recessive, autosomal dominant or autosomal recessive. Suppose color blindness, whether it is autosomal dominant or autosomal recessive, or myotonic dystrophy that pedigree chart has given in your NCERT books, whether it is autosomal recessive or autosomal dominant. Sickle cell anemia, autosomal dominant or autosomal recessive. This one caused by the point mutation. I think you know. Now we are going to see some diseases. Let me tell you some autosomal recessive, autosomal dominant, which are actually comes under Mendelian genetic disorders. And after that, we'll be telling about the sex-linked dominant and sex-linked recessive. As I have told you earlier, that Down syndrome, then Klinefelter syndromes, then Patau syndrome, Edwards syndrome, Pratt-Whitney syndrome. These are actually not comes under Mendelian disorders. These disorders you cannot analyze or rule out the patterns of the disease when it will arise. Cannot find out. So this is actually one kind of drawbacks of the pedigree analysis, or you can say limitations of the pedigree analysis. The question will come. Now let's see 2 or 3 examples. Recessive only I am going to write autosomal recessive, autosomal recessive. So let me give you some list so that only we will use uh, during pedigree analysis, so that you may not having any issues to find out which side is going to take. First one you can use as, we'll start from A alphabet, L captonuria, L captonuria, captonuria, sorry, captonuria, captonuria. So which will cause due to the defects of chromosome number 3. Second one you can use cystic fibrosis, cystic, what I told, fibrosis. This will cause, I think, maybe chromosome number 7, you can check. Then phenylketonuria, this is very very important. On this topic also I have given one videos, you can check. So you can use PKU, I think it will be chromosome number 12. This will be chromosome number 3 probably, you can check chromosome number 7. Then one of the most important is what, sickle cell anemia, sickle cell anemia, sickle cell anemia, which will be caused on chromosome number 11. Okay, so it is quantitative or qualitative. This is question 1, this is for your homework. Quantitative or qualitative, you just give me the answer in the comment box. This is for your homework, very very important. You need to know the concept about the quantitative and qualitative. Then another thing which is very very important again, which is called as thalassemia. Thalassemia, it is also one kind of what, it is also one kind of autosomal recessive. So there will be two types of thalassemia. One is alpha thalassemia, another one is beta thalassemia. Alpha thalassemia will be occurred maybe in chromosome number 16, and this one may occur in chromosome number 11. You should know at least the chromosome number, because question may come. Another one which is actually not including in your NCRT, which is, you can, some questions may come during pedigree analysis in NEET exam, albinism, mainly on chromosome number 11, but if we tell only chromosome number 11, it will be misleading, but mainly on chromosome number 11. Multiple chromosomes may be affected. So you have seen chromosome number 3, chromosome number 7, chromosome number 12, chromosome number 11, 16, 11, 11. These are actually autosomes. So that is what comes under autosomal recessive disorder, which are actually called as what? Which are actually comes under Mendelian disorders. Now in case of this one, autosomal dominant, let me write autosomal dominant, autosomal dominant disorder. I have taken only two. One is Huntington's disease. One is what? Huntington disease. It may be, I think, chromosome number 4 will be affected. Another one which is given by NCRT, the pedigree I have seen. This one is called as what? Myotonic dystrophy. So tonicity, muscle tonicity will be affected. While it will be relaxed, it cannot be pull. Okay, myotonic dystrophy, DYS. By the name itself, you can write what is myotonic dystrophy. Myotonic dystrophy. It will be maybe chromosome number 9. So these are actually, this list you're supposed to memorize. This list you're supposed to memorize. If question will come, question directly will not come that which type of disease, whether it is autosomal recessive and autosomal dominant. Question will come. I'll come to know. You can find out the pedigree analysis of the given chart, and which of the following disease or which of the following disorders will be caused by this pedigree. Options are alkaptonuria, Huntington's disease, hemophilia A, color blindness. So many options they will give. So obviously they have given 4 options. So under the 4 options, if they will mention the disease or syndromes names, so you cannot identify which is perfect. Although if you have knowledge on pedigree analysis and all, but you should remember all the disorders name. Otherwise, you cannot find out or tick the exact options in exam. Okay. Now let's list some sex-linked disorder. Sex-linked. So sex-linked disorder will obviously, probably you may recall, is supposed to be X-linked or Y-linked, isn't it? So we'll be giving preference on the X link, okay? Because in case of Y links, we know the Hollandric genes that you know about the hypotrichosis, which is the arising of excess hair outer ear of pinna. So you have seen this will be passing from father to fathers. These things actually I'm taking it aside now, which is very important for the NCRT and the need purpose. You should remember at least 5 to 6 numbers of diseases which are called as autosomal dominant and autosomal recessive. First one, let me write sex-linked dominant. Sex-linked dominant. Sex-linked dominant. You can write Rett syndrome, or you can write hypophosphatemic ricket. Hypophosphatemic ricket. So this is, you have seen hypophosphatemic. If you break it, then you will be getting, there will be less of phosphate or release of phosphate through the urine, and due to this also that bone formations will be having some issues. Also here vitamin D deficiency you can use. So what it is, or vitamin D resistance also you can use, which is called as hypophosphatemic disease or hypophosphatemic rickets. So that rickets is not that rickets which will actually we have heard in the nutrition chapter, isn't it? But this is actually genetic disorders, hypophosphatemic rickets. These two rickets are different. Okay. Then autosomal recessive, which is very very important. You can use another one, Rett syndrome also. RD double D, Rett syndrome. These two syndromes actually out of the syllabus. If somehow they will give X-linked dominant, without mentioning X-linked dominant, they have supposed to used hypophosphatemic rickets in the options. So in options, if you will see that type of options without knowing, you may click in the exam fear conditions. So obviously you should at least know the name. I have actually listed 25 numbers of disorders. At least 25 numbers of disorders you should know. Then only you go for pedigree analysis. Okay. Now X-linked recessive, which is very very important. Recessive. All the students maybe, or most of the students will be giving preference to autosomal recessive and autosomal, sorry, X-linked recessive and autosomal recessive. But you should know at least two or three disease name in case of autosomal dominant and X-linked dominant. X-linked recessive, which is very very important, hemophilia A, hemophilia B. You can use hemophilia. So I'm not going to explain the depth of the hemophilia. What is A hemophilia? What is B hemophilia? Another hemophilia also there, which is called as hemophilia C. Or my concern is to know about the, or to explain about the disease name. Okay. Then another one is what? Color blindness, which is actually called as red color blindness. Sorry, red green color blindness. So then will be what? Red green color blindness. So obviously, by the name itself, the sufferer cannot identify which color is red, which color is green. Cannot identify, or they cannot. This disease is also called as daltonism, on the name of the scientist who actually discovered. Then another one is muscular dystrophy. Previously, I have used myotonic dystrophy. Now I am using muscular dystrophy. You can discover muscular dystrophy. So these are the listed. These are the list of the diseases which I am going to find out through the pedigree analysis. In which generations, the charts I am going to know. The charts I am going to explain. Okay. So these are the disorders. Their disorders are called as, or are comes under Mendelian disorders, because here I have used recessive, I have used dominant, I have used recessive, I have used dominant. So dominant recessive, while I will use, or you will see anywhere, then without knowing anything, you just write it is comes under what? Mendelian disorder. Not other chromosomal disorder. So other chromosomal disorder in NCERT it is written what? Chromosomal disorder it is written, which is you supposed to know, or better to use non-Mendelian chromosomal disorder. Non-Mendelian, what I used? Non-Mendelian chromosomal disorder. Okay, now let's start one or two pedigree. Okay. So let's identify what alleles you are going to use. As A is starting from the very beginning, so we are going to start from the A alphabet. So autosomal disorders. So that autosomal disorders will be classified after a few minutes ago what I told, dominant and recessive. So what type of alleles you are going to use? In case of autosomal dominant, let me write capital A, capital A, capital A, small a, small a, small a. See here, if it is autosomal dominant, if it is autosomal dominant, while you will see capital A, red color I am going to use, which is diseased person. Obviously that person is diseased or affected. If also you will see capital A small a, then also you can use or think that that person is diseased or affected. And in case of small a and small a, if it is autosomal dominant, then you will use it is normal person. So you have seen in case of autosomal dominant, I have not mentioned any carrier. Okay, so there will be no carrier of autosomal dominant disorder. Now here also I have not used the probability of having the autosomal dominant disease or disorders on XX or on XY, that is on male and female. So as it is not X-linked, so you cannot, or you don't need to worry about the probability of having male and female. Now in case of autosomal recessive, same things I am going to use capital A, capital A, capital A, small a, small a, small a. As it is autosomal recessive, this one will be what? Let me write autosomal recessive, REC, autosomal recessive. So this one will be what? This one will be diseased. Clear? And capital A, small a, you have seen this one I am going to use that green color. This one will be normal, but you can use carrier. Another term what I have used, carrier. So autosomal recessive disorder will be carrier, will be carried by some persons. So capital A, what will be there? That capital A, capital A also in case of autosomal recessive, we are going to use as for normal person. So you have seen how difference, what are the difference between this one, autosomal dominant and autosomal recessive. Clear? Now let's come to the X-linked dominant and X-linked recessive. Okay, X-linked, red I'm not going to use, we'll use different color. Let's see, X-linked, X-linked. Here also we'll start from the dominant part, because dominant we should give the preference to the dominant, we will start from the recessive. Otherwise, because you need to know here in the Mendelian chapter, so dominant will be dominant, isn't it? So it's supposed to be started from the very beginning, so it is dominant. And here will be what? Recessive. Clear? Do not do hurry, otherwise it will create confusion. Now let me write, suppose x x x x x x. I will use only 5 times. Here also x x, sorry, xy and xy. Here also let me write x x x x x x xy xy. Now in case of this one, you can use capital alphabet T, or you can use the circle, which is the older method. In college also this method we have used to identify which is X-linked dominant, which is X-linked recessive. See, suppose here XX, suppose it is not having anything, XX. That XX is what it is, female. It is actually, you can write normal female. Clear? So that box you cannot use as per pedigree. Let me rub it, otherwise misconception will be creating in case of you. Right? Now see that if you will see X, this one also you can use, or this one also you can use. Any alphabet you can use to identify the pedigree or the consent. This one will be what? X0 I have used, X0. X0, this will be what? Diseased. This will be what? Diseased. It is diseased, or you can have DF, diseased female. So if you will see 2 dot, 2 dot, or you can use 2D. Then you can use what? This is also what? This is also affected female or diseased female. In case of this one, if we will be having XD or X0, it will be what? It will be affected or diseased male. Diseased male. This is autosomal dominant time, sorry, X-linked dominant time. And in case of X, obviously it is what? It is what? It is normal male. Clear? Okay, now X-linked recessive. In case of X-linked recessive, obviously see, if you will see, let me take another color. If you will see, suppose capital R, capital smaller I am using, smaller, small, it is not visible properly. Let's write small r. Or you can use round, because this round will clear you so many confusion. Smaller, smaller. I have given capital R. Smaller. If you will see this one will be normal female. This one will be what? Normal female. Normal female. Or you can use, yeah, normal female you can use, or you can use carrier female also. Yeah, if you will see nothing on X, this also again normal female, but it is not carrier. C also. And if you will see uh, small r or X0 X0, then it will be what? Then it will be affected female. Then it will be affected female, or DF. You can use DF. This is female. Now here what it is? XY only written. What it is? XY. It is what? Normal male. If you will see here see, if you will see another very interesting, you will see X0 or XR smaller, then it will be having what? It will be diseased male. It will be what? Diseased male. Now see, in case of female, that females are the carrier. Okay, in case of X-linked, the females are the carrier, but males are not the carrier. Males are mostly affected. You have seen here also it is affected. Here also you have seen affected. No carrier. Here you have seen there will be the carrier. So these things you have to know. which type of alleles you are going to use, then only you can rule out the pattern of pedigree. Now let's start what given in the NCERT. Two pedigree they have given. After that, we'll explain the many more. We'll see a lot of things. Before going to start the pedigree analysis, let's see some trick. Okay. So first trick you can use for dominant. This, if you will find out from the very beginning, your work I think 50% done. Dominant versus recessive. Clear? So if you will see any kind of pattern of the disease which is actually does not skip the generation, does not skip the generation. We'll give you through example after a few minutes. Just wait. Does not skip the generation. Then it will be what? Then it will be comes under dominant. Does not skip the generation. Means in every generation you will see any affected persons. Okay, F1, F2, from parent to F1, F2 you will see. Then this will be dominant. If it will skip, if it will skip, if you will see the skip, then it will be recessive. So then obviously the 50% of the pedigree you have completed. Now question is, this one actually depending on the pattern I have made, then whether it will be autosomal or X-linked. It is dominant and recessive. Now let's do the war between autosomal versus X-linked. Clear? In case of autosomes, but you will see some pedigree that pattern will not follow. That is why actually I am preferring my students do not follow the pattern, do hard work. Then that hard work will give you some best fruits for your life. See, autosome versus X-linked. If it is autosomes, both male and female are affected almost equally, but there are so many pedigrees. But here we'll see in case of X-linked also male and females are affected almost equally. Clear? So male and females are affected equally, then it is comes under what? It is comes under autosome. And X-linked what? X-linked actually depends on sex. X-linked pattern depends on what? Depends on sex. Okay, let me give you something more in case of X-linked pattern. If it is suppose X-linked dominant pattern, if it is suppose X-linked dominant pattern, then what will happen? You will not see the transmission from father to son, you will not see. So that means what? It will be cross. You will not see. You will see affected father. Affected father, that is red color I am using to for affected father. Affected father passes the disease to all daughter. Clear? To all daughters. So you have seen father to daughter. And you will not see no female carrier. You will not see no female carrier in case of X-linked dominant. You just think about this one, what I have given on the previously. You will not see no carrier female. No carrier female. Okay. And X-linked recessive. Let's start about the X-linked recessive. These are actually the tricks to do the pedigree in first way. In case of X-linked recessive, mostly male will be affected. Males affected. Okay. It does not mean that the female cannot affected. Mostly males are affected. You will see females are the carrier. Females are the carrier. Okay. Females are the carrier. But you will not see the males are the carrier. Okay. Females are the carrier. And also another rules you can use, no father to son transmission. No father to son transmission. So here also you can use father to son transmission, not seen. Okay. So these are the tricks you can use to analyze the pedigree. Let's start from the NCERT. From the NCERT, I have taken this peak. Now, can you identify any pattern here? You have seen that both the mother and father are not affected, but their boy or their son and daughters are affected. Here also you have seen This one is not affected, but their boys are affected. At as I have told you, if you will see that, as I have told you earlier, you will see that pattern. See here, the mother and father are not affected, but here you have seen the son and daughters are affected. Also here, son is affected. Now that means what? You can assume that it is concerned or recessive, isn't it? Because here it is missing. The uh, generation is here. See, the disease is missing. So now question is whether it is autosomal recessive or X-linked recessive. What I told, if in case of any pedigree, both the male and females are affected equally. What I told, almost equally, isn't it? Almost equally. So it cannot consider X-linked. It will be what? Autosomal recessive. Now, so let's assume that this is autosomal. This is now you have I think assume that 70% assured that this pedigree is autosomal recessive. So let's prove it by doing the crossing, isn't it? So in case of autosomal recessive, so what's supposed to be allele? Capital A, small a. It will be carrier man, maybe. Here also capital A, small a. You can take this one also. Capital A, or capital A, capital A. Now question is what will you take? Okay. Now see, if you take this one, capital A and capital A in case of male and female, obviously one supposed to be carrier. One supposed to be carrier. Otherwise, how that disease will cause to their progeny, isn't it? So suppose you can make carrier to the female. Do not think in another way. I have made the carrier to the female. Okay, you can make the carrier to male as well. I am making the carrier to the female. Okay, so capital A, small a. Now question is why are supposed to be in case of male, or you can use as a in case of father. Suppose you have taken this one, capital A, small a. So what will be the probable genotype here? Capital A, small a, isn't it? Here also you can use small a. Here you can use capital A, small a. Here you use small a, capital A, small a. Now that lady, which is carrier, he actually married with a normal person. Suppose married with a normal person. I have taken capital A, small a. So what will happen? Then one will be small a. Here you can use capital A, small a, and here you can use capital A, small a. Now I have taken what? I have taken capital A, small a. This one. Now you may ask some questions, sir. Why not capital A? Capital A you have taken. See, capital A, capital A. If you will suppose this one I have taken, cross to this one, what will happen? Capital A, small a, and here it is okay, capital A, small a. But here what will you do? Here the one allele supposed to come from the mother and another allele supposed to come from the father. Then only this one will become affected. But here if you have taken capital A, then it will be carrier. But it's supposed to be resist. That is why you cannot take capital A, capital A. Okay. So that is why you cannot take capital A, capital A. The genotype will be capital A, small a in case of father and capital A, small a in case of mother. That means what? Both the parents are carrier. Both the parents are carrier. Isn't it? That means what? Both the parents are carrier. So this is actually called as what? This is actually proof that this one is autosomal recessive trait. 100%. Clear? Now let's do the another one which is given in the NCRT. Now let's check the pattern. Here you have seen the affected parents. Here you have seen that affected progeny. Here you have seen that affected progeny, which is girl, which is boy, and also it is what? Again, girl. Also another thing also you have noticed or not, this disease is transferred from mother to son, but here it is transferred from uh, mother to girl. Now question is whether it is dominant or recessive. First you need to identify. You have seen in all the generations. Here it is parental generations. You have seen one affected. Here also you have seen one affected. Okay, so okay. So here after that one, this one when they have got married, another also one will be affected. Here what happens, sir? Here as they are not affected, offsprings are not affected. You just keep it in your mind. So in one sense you can write it is actually a dominant property. It is actually dominant. It is actually considered dominant. Now question is whether it is X-linked or autosomal. It is autosomal dominant or X-linked dominant. Let's do in two ways whether it is finding autosomal dominant or X-linked dominant. Okay. In some pedigree I have seen both the traits are or both the diseases are seen. Autosomal dominant also, X-linked dominant also in some pedigree I have seen. But let's see whether it is what results findings. what it is, I have left it as A started. Obviously, I am giving to preference the A. So here it is autosomal dominant. Obviously, it will be what? Capital A, or you can use capital A, small a. Whereas it is a normal person, obviously, you're supposed to use small a. See, let's do the cross. Suppose you have taken capital A, small a. If they will ask the genotypes, then what will you do? Obviously, you have to mention the exact genotypes. Capital A, small a. Here, what? It is normal. So, in normals, here you're supposed to use small a. So, that means what? Capital A, small a, you have to use. Otherwise, it cannot be a normal person. If it is capital A, in case of autosomal dominant, obviously, it will be affected. So, you can run this one. So, exact genotype will be what? Capital A, small a. What will be the probable genotypes? Capital small a. Here will be what? Capital small a. Here it is normal person. So you can use small a. Here again capital small a. These things I think clear. Now this normal boy married with a normal girl. Suppose small a. What will happen? All the progeny will be small a. Clear? Now this is affected, which is autosomal dominant. Married with what? Married with normal boy. Now what? My battery is going to die actually. Now what will happen? This is normal person. This is normal person. You can capital small a. It is affected. Capital A, small a. Here also capital small a. That means what? It is actually in one way. It is actually proved that it is autosomal dominant, isn't it? It is autosomal dominant. Now let's put whether it is autosomal dominant. It is already proved. Now whether it is X-linked dominant or what? Why not it is X-linked? So let's prove X-linked dominant. Clear? This one autosomal dominant is proved. No confusions. Now let's see whether it is X-linked dominant or not. Let's wrap all the things. Now as it is X-linked dominant, obviously it will be what? X, Y, normal person. X-linked dominant. There are some carriers also. You have to use X dot X. Clear? You cannot use X dot X dot. I will use dot only. I am not using any alphabet. Okay. So these things why cannot sir use? Because if you will do the cross here, you can randomly cannot take capital A, small a. Here you're supposed to do the cross. Here you're supposed to do the cross. What? Now how many numbers of progeny here form? 1, 2, 3, 4. So let's do the cross. X dot X cross with X Y. So X dot X, X dot again Y, X, 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 Y, X, Y. Now see X dot X. That means what it is? Affected. X dot X again. X dot X. Again it is affected. X dot. Sorry, this one will not be X dot X. This will be X dot Y. X dot Y. That means Y is affected. Now XX. XX. Now X Y. So you have seen 50% of the offsprings are affected and 50% offsprings are not affected. So you have seen there are 4. If you will correlate with this one, you have seen X dot X. So what will be there? This one will be what, sir? X dot X. And this one will be what? This one will be XY. This one will be X. Sorry, this one will not be XY. This one actually in the married persons. You cannot write this one. This one will be XY. This is suppose was XY. This is normal person. Now let's take X dot Y. X dot Y. This is actually disease person and XX. 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 So see, till now it is fitted into the X autosomal dominant disorders. Now let's see next generations. Next generations. Okay. So what can we do? You have to skip this one. You have to skip this one. Why this only you cannot do? Okay. If you do the cross, then only you have to find out. Now what will happen? This is normal boy and this is also normal girl. So here what will happen? This is supposed to be X and X. X and X it will form XY, XX, XX. Perfect. Now let's come to the final one. Here it is supposed to be XY as it is autosomal dominant. So let's do the cross like the previous one. X dot X already done. X dot X, X dot X, XX, XY. X dot X, XY. Okay. Now let's do. So what will happen that X dot X, X dot X, X dot X. It is okay. But here any one of the boy here, any one of the boys supposed to be a diseased. Here you have seen one boy is diseased, one boy is diseased, one boy is diseased, another boy is not diseased. It is okay, no problem. You can write XY. But here it's supposed to be X dot Y. X dot Y on the basis of this one, but it as what? It is XY. So here what will happen? Here it cannot be autosomal dominant. It will be sorry, it cannot be X-linked dominant. It's supposed to be autosomal dominant. Understood? Let's do the another pedigree, which is not in the NCRT, but the theory it is given in the NCRT. Now let's find out whether anything will be ruled out or not. It is for this only you have to think beyond. See, this is the affected female. That affected female, I'm going to use red color. That affected female had married with a normal boy. Then all the progenies are affected. Oh my god. After that, what will happen? That boy, that affected boy, married with a normal girl. But here, no progenies are not affected. But here, what happened? That affected girl, while married with a normal boy, then again same properties you have seen. All the progenies are affected. Now what will happen? You can see Now you just check the trick. That is why I'm not giving the preference for giving the trick to the student. Now see, in all the progeny that suppose this is P1, P, this is F1, first filial, and this is second filial, F2. Here all the offsprings and the members are affected. So that's supposed to be what it is dominant. As what I told, as it is equal, the affected will be equal in case of male and female. Obviously, it cannot be X lineage. It's supposed to be autosomal. So the answer is autosomal dominant, but it is totally wrong. So their here tricks not work. So the answer is what? The answer is cytoplasmic inheritance. The answer is cytoplasmic inheritance. Inheritance. So you have to know about the cytoplasmic inheritance. What happened? So you know about inside the cell there will be the mitochondria. That mitochondria will be having some DNA. That DNA is actually have that circular DNA. The DNA is helping in the what? The DNA is helping in the transmission of the inheritance from all the progeny. As you know about the reproductions, already you have finished. I think there will be the nucleus, there will be the sperms. From the sperms only, that DNA content will be fused with the nuclear content of the eggs. Also, there will be what, some cytoplasmic content. In the cytoplasmic contents, also what will happen? That mitochondria, it's a DNA. So this one ultimately enter, it will form what? Zygote. So that means what? The cytoplasmic content of the zygote will coming from the mother, not from the father. So you have seen that female who is affected, he actually passing the traits all the progeny, all the progeny. That is why you cannot write it is autosomal dominant. It's supposed to be cytoplasmic inheritance. Understood?